Mitochondria are the engines of our cells, the critical sites of energy production, oxidative balance, and metabolic control. As we age, these organelles accumulate damage from reactive oxygen species, ROS, experience declines in efficiency, and begin to falter in their energy-generating capacity. This mitochondrial dysfunction is a central feature of aging and a driving factor behind age-related decline in tissues with high energy demands, such as the brain, muscles, heart, and immune system. Fortunately, growing evidence suggests that both dietary carbohydrate restriction and targeted vitamin intake, particularly B vitamins, can work in tandem to support mitochondrial health, preserve cellular energy balance, and slow the pace of physiological aging. The metabolic stress that comes with aging is partly fueled by overnutrition and chronic carbohydrate overload. Diets rich in refined carbs and sugars keep insulin levels elevated, which not only impairs metabolic flexibility, but also overwhelms mitochondrial function. Chronically high glucose exposure leads to excessive RLS production, glycation of mitochondrial proteins, and impaired fatty acid oxidation. As a result, mitochondria in older adults often struggle to switch efficiently between energy sources and become less responsive to signals that would normally stimulate repair or renewal. Over time, this contributes to fatigue, cognitive decline, insulin resistance, and inflammatory diseases. Low-carbohydrate diets offer a way to reverse many of these effects by relieving mitochondrial stress and enhancing metabolic flexibility. By reducing carbohydrate intake, insulin levels drop, promoting lipolysis and enabling cells to rely more on fat oxidation and ketone production for energy. This metabolic shift not only reduces ROS generation but also favors mitochondrial biogenesis, the creation of new, healthier mitochondria. Ketones themselves, especially beta-hydroxybutyrate, act as signaling molecules that improve mitochondrial efficiency, activate protective genes, and promote autophagy, including the selective removal of damaged mitochondria, mitophagy. In this state, energy is generated more cleanly and more sustainably, supporting both cellular longevity and whole body health. B vitamins, especially B1 thiamine, B2 riboflavin, B3 niacin, B5 pantothenic acid, and B7 biotin, are essential cofactors in mitochondrial energy metabolism. These vitamins form part of the enzyme complexes that regulate the Krebs cycle, fatty acid oxidation, and the electron transport chain. Without them, mitochondria cannot efficiently convert macronutrients into ADP, the cellular energy currency. For example, thiamine is needed for pyruvate dehydrogenase, a key enzyme linking glycolysis to mitochondrial respiration. Riboflavin and niacin contribute to the formation of FAD and NAD, electron carriers necessary for oxidative phosphorylation. Deficiency in any of these vitamins can lead to fatigue, metabolic bottlenecks, increased oxidative stress, and accelerated cellular aging. Among these, niacin and its derivatives, NAD, precursors like NMN or NR, are particularly important in aging. NAD levels decline with age, compromising the activity of sirtuins, a family of enzymes involved in DNA repair, inflammation control, and mitochondrial maintenance. Low NAD also impairs autophagy and mitophagy, leading to the accumulation of dysfunctional organelles. Supplementation with NAD boosters has been shown in animal models and early human studies to restore mitochondrial function, improve insulin sensitivity, and enhance endurance, all of which contribute to healthier aging. When combined, carbohydrate restriction and B vitamin optimization create a metabolic environment highly favorable to mitochondrial preservation. Low-carb diets provide the substrate pressure that forces mitochondria to function efficiently, while B vitamins ensure the availability of enzymatic cofactors needed to handle the increased load of fat oxidation and ketone metabolism. This dual approach reduces metabolic stress, suppresses pro-inflammatory signaling, and enhances the cleanup and regeneration of damaged cellular components. Additionally, mitochondrial biogenesis is promoted by key signaling pathways such as AMPK and PGC-1A that are activated under energy stress and supported by nutrient sufficiency. Low carbohydrate intake naturally raises AMPK activity by mimicking energy scarcity, and B vitamins, particularly niacin, ensure adequate NAD for sirtuin activation, which enhances mitochondrial gene expression. These pathways are not only critical for energy production but also for defending against age-related diseases such as type 2 diabetes, cardiovascular disease, neurodegeneration, and sarcopenia. Unlike calorie restriction, which can induce nutrient deficiencies if not carefully managed, this dual strategy offers a more sustainable, nutrient-replete model. Carbohydrate intake is lowered, but not eliminated, favoring slow-digesting, fiber-rich, low-glycemic sources. Protein and healthy fats are maintained to preserve muscle mass and hormonal balance. Micronutrient intake is optimized, either through diet or supplementation, to ensure mitochondrial enzymes and antioxidant systems remain fully functional. In summary, supporting mitochondrial health is not just about reducing calories or taking a single supplement. It is about creating a coordinated dietary environment that minimizes metabolic overload while supplying the cell with the precise cofactors it needs for energy and repair. Low-carbohydrate diets reduce the burden on mitochondria and encourage their renewal, while B vitamins enable their full performance and resilience. Together, they represent a powerful, accessible strategy to combat age-related decline, optimize energy, and promote long-term vitality. As we continue to uncover the intricate relationship between diet, micronutrients, and cellular aging, this dual-focus approach offers a promising foundation for mitochondrial longevity and healthy lifespan extension.